Good morning, all of you. At the outset, I thank the Director of Public Health, Dr. Selva Vinayakam, and his team of the Department of Public Health for having given me an opportunity to talk on it in an issue which is Omics technologies, which may be slightly high sounding at this point. But once I get into the discussion, you will understand how much it is very close to the public health and the public health specialists in terms of ground management in the community. The session that I would be running I will be going through what are the different types of omic technologies. That is the long read sequencing and the spectroscope based omics, application of genomics in the public health in terms of population risk and population scale pharmacogenomics, and of course a few examples in which I have been a part. So before I get into omics, I need to put in order certain basic issues in public health in the current context. Non-communicable diseases and chronic debilitating disorders are slowly moving into the center stage of public health with the reduction in communicable diseases and those smaller entities which have been commanding the center stage for all these years. When you come to Europe, currently 2/3 of the deaths and 75% of the healthcare costs is because of chronic disorders. And this chronic disorder is of huge economic impact that it actually consumes a lot of the budgetary allocations in these countries. But that is not the way a healthcare can run in the coming years. So, attempt to solve these problems at an earlier phase or in a previous level than it is being done presently has to be thought of. In the US, if you do a hot mapping of chronic disorders, you find that
I request all of you to keep silence, please. If you have anything to talk, you can go out and talk. While leaving also, please maintain a silence. Please, if you have anything else, you can just go out and do it. So discipline is first, please.
Thank you so much. Yeah. So when you come to the United States, we are seeing a very interesting phase where the United States has transitioned into a phase where the predominant disease the public health in the US is facing is chronic disorders, especially the non-communicable diseases and other chronic degenerative disorders, especially the neural and the cardiac and the hepatic disorders. So the public health there has a new issue where the highest prevalence is facing socio-economic and environmental barriers. This has to be put in mind because we are not transitioned, but we are in the phase of being transitioning, right? So now let us come to our own public health. Years before, the public health was battling to reduce deaths due to pandemics. Slowly it moved to a phase of surveillance where early identification of infectious diseases reduced deaths and morbidity. Then came the phase where vaccination was introduced and vaccination reduced the occurrence of a lot of infectious diseases. Then came nutritional and health support for the mothers and the newborn. So what happened? Maternal and infant mortality reduction was our phase. Then presently, with what we call as the Makkalai Thedi Maruthuvam and also the NCD program of the Government of India, we are now focusing on early identification of non-communicable diseases, reducing the complication rates in the community by survey and health support. But preventing NCDs, early identification of chronic debilitating disorders, and preventing chronic debilitating disorders is not in our framework or in our scenarios at this present moment. What do we do about it? See, basically, I think I will go back to a reference of the WHO where it says chronic diseases are of immense and importance and increasing the toll of lives in the community. We now have much better technology available like risk prediction tools, but we are not systematically implementing them at the required place, is a recommendation of the International Common Disease Alliance for Polygenic Risk Scoring. It's a task force which has specifically said that the public health needs to use better technologies in risk prediction. And one of those technologies which I am talking about here, that is the Omics technology, is going to be a huge tool in public health. So what is Omics technology? Omics technology is a comprehensive biological data. It's a comprehensive, large biological data which comes from major 5 types of omics. That is genomics, proteomics, metabolomics, and transcriptomics. There are 2 other smaller omics, that is the microbiomics and immunomics. They are adjuvants to the whole issue. But we need to remember genomics, proteomics, metabolomics, and transcriptomics. These 5 issues will be driving the public health effort in the next 10 to 15 years. Now, I will go through the process. This omics is a new terminology. I will put it across to you. You take the cell. The center of the cell, you have the nucleus, which has the DNA, which decides what the cell will do in terms of its participation in the human body. So, the DNA is composed of smaller molecules, which we call as the base pairs. So, that globality is called as genome, and the study of it is genomics. When the DNA transcripts or tries to produce proteins, it converts into mRNA, and the studying of all the mRNA in one go is called as transcriptomics. When the mRNA reaches the endoplasmic reticulum, it produces peptides, polypeptides, and proteins, and that entire community of chemicals that are produced is called as the proteome. These proteins structurally and functionally get involved with the metabolism of the cell and produce a lot of smaller products because of the metabolism of the amino acids, the glucose, the lipids, and the nucleic acids. Those molecules together are called as the metabolome, and study of it is called as metabolomics. Now applying this to a timeline from a defect in the cell to a clinical diagnosis of a disease, you will understand that within nanoseconds of a defect happening in a cell or in a nucleus, you can diagnose it if you do a genomic sequencing of the nuclear DNA within nanoseconds of onset of a disease. Nanoseconds to microseconds of the onset of a disease, you can diagnose by finding out the changes in the mRNA or aberrant cell signaling. Microseconds to few seconds, you will see the cells producing aberrant proteins. Then seconds to few hours, the cell will start producing aberrant metabolic products or aberrant cell function will start. And few days to few years, there will be structural and functional changes that the cell abnormality will cause, which will be clinically diagnosed. If you look at this picture, you will understand that the present day public health is relying on clinical, imaging, and laboratory data for early diagnosis of diseases, even for preventive strategies. So, we are using only 1/10 of the data of a human disease process in deciding what the public health intervention needs to be to prevent diseases and to provide a healthy society. So the responsibility of the public health now is to harness the large unharnessed data that lies in front of us in terms of omics or omics-based data or omics-based technology-based data. Now I will look into what is the so what is the unharnessed data we have? Take genomics. You have data about 300 million base pairs. Transcriptome gives you 1.5 lakhs data points. Proteome gives you 20,000 data points. Metabolome gives you 2,500 data points. And these data points can be derived using the same material that you are using for the present day clinical diagnosis. But only one thing, the diagnostic technique requires certain specific platforms, which is called as the next generation sequencing or the third generation sequencing, and of course the mass spectroscopy. These are two techniques which can add on to your data. Now, the question comes, is the public health capable of doing it? You should understand that without actually knowing that you were using omics data, you have gone through the process of controlling a pandemic in the last 2 years, where wonderfully you have used omics data of the SARS-CoV virus to actually in January 2020, that is when the first entirety of the SARS-CoV virus was sequenced. In March of 2020, you had the detection kit, which was a prototype sent into for manufacturing. By April, the public health community of the world had produced the prototype of a vaccine, an RNA-based vaccine, and by June or August, it was already under production. And January of 2021, all of us had the first shot of a corona vaccine. That is all because of the use of omics technology by the public health. This is one classical example. And so, omics technology is not new to public health, but there are certain other uses which we have not thought of. Now coming to, I will just go through a few slides where I will describe what these omics technologies are. Number one, the genome sequencing for DNA and the mRNA. In 2003, these slides are here to make you understand how it is easy for you to use omics technologies in public health in the coming years. In 2003, the first human DNA was sequenced in the National Institute of Health in the US by a US scientist called as Craig Venter. He had spent 13 years and $3 million and 400 Sanger sequencer machines to produce the data about one human genome. That is 13 years. 2009, 6 years later, the first Indian human genome was sequenced by the CSIR Institute of Genomics in Delhi. And this was in 50% of the time that the US had taken. In 2020, India had already sequenced 1,000 Indian genomes, which is called as the Indigen program of the government of India. By luck or whatever, I was part of the 50 scientists who were involved in the sequencing of the first 1,000 genomes in this country. Now, you look at this. When Venter did it, he spent $3 million for sequencing one human DNA, human gene, sorry, human, the 23,000 genes. But today in 2022, you can do the same job for less than $400. And what machine which we were trained in genomics is a machine which is almost filling 1,000 square feet space. But today, you have sequencers which use what is called as the nanopore sequencing technologies. All of these in the picture are all handheld machines. You should remember, these are all handheld sequencers which the public health people can carry in their hand when they walk into the community to do sequencing on ground. So now coming to the others, that is mass spectroscopy in metabolomics and proteomics. You have the what you call as the liquid chromatography, which was introduced in 1987, which is one powerful machine available in many hospitals in this state, which can do a proteomic study. Similarly, a machine which is called as a gas chromatography and other types of machines, which produce data on metabolomes, are also available in our own system. So our system has the machinery and capability today to do proteomics and metabolomics. With a short reference to microbiome, microbiome is the sum total of the physiochemical activity of the microbes that are there in all of us inside our gut. Very interestingly, the amount of the microbes that we have in our gut is so much that you can call it as the second genome, and the physicochemical activity of these microbes alter the body in terms of our disease responses and even our disease risk. So this is one thing that could add to the data of the public health. Now coming to this. See, you have almost 3 million base pair variables. You have 1.5 lakhs of transcriptome variables, 20,000 odd proteome variables, 2,500 of metabolome variables, and 200 to 500 of clinical variable data for almost every patient in the field. You should understand what amount of big data the public health has, the amount of computational skills that our country has put into use. This will give you so much amount of preventive and predictive value that 10 years down the line, 15 years down the line, what is going to be the state of the population which you are handling in terms of risk and risk attenuation, it would be very easy to sit in your offices and predict and prepare the health system to handle them. Now coming to a very interesting law which is called as the Moore's law, which has been used in computational power in the recent years. I reiterate the same law here. We have two disruptive technologies that we have here. One is the computational power in terms of big data analysis and data handling. You have another big is the Omics technologies. These two disruptive technologies are there in the doorstep of the public health today. If you can put them into your control, into your use, the power of the public health or the disruption it will cause, I will tell you one thing, it's not for a joke. The importance of the tertiary care, as many of our speakers were also mentioning, the importance of the tertiary care will dwindle down in the coming years because the public health will be able to prevent diseases, prevent risk, prevent complication due to risks at a very high level that use or requirement for large hospitals with large capacities and large infrastructure may not be necessary in the coming 10 to 15 years. Now coming to what are the current application of omics technologies, I'll go through you very rapidly. Cancers and chronic diseases is going to be the place where these omic technologies work in terms of population risk. I will but mention some of them. Beta thalassemia. I think many of the people in the public health who are in these districts. Yeah, please.
See, டெக்னிக்கல் செஷன்றது கேட்கறதுக்கு தான், பேசுறதுக்கு இல்லை. சரியா? நான் மறுபடியும் ரிமைண்ட் பண்ணிட்டு இருக்க மாட்டேன். சரி, நம்ம அவர் என்ன விஷயம் சொல்றான்றது தயவுசெய்து தெரிஞ்சுக்கோங்க. உங்களுக்கு இந்த செஷன் பிடிக்கலன்னா சத்தம் இல்லாமல் வெளியே போய்ட்டு ரிலாக்ஸ் பண்ணுங்க. தயவுசெய்து மற்றவங்கள டிஸ்டர்ப் பண்ணிட்டு இருக்காதீங்க. சரி, ஒவ்வொரு ஸ்பீக்கரும் எவ்வளோ எஃபர்ட் எடுத்து ப்ரிப்பேர் பண்ணிட்டு வந்திருப்பாங்க உங்களுக்கு தெரியும். சரி, சயின்டிபிக் செஷன் ஷுட் பி எ சயின்டிபிக் செஷன். நேத்திக்கு கேலா, சரியா? இன்னைக்கு 6 மணிக்கு வாங்க, சத்தம் போடுங்க, டான்ஸ் ஆடுங்க, தட் இஸ் டிஃபரெண்ட். Scientific session should be treated science. Please.
Thank you, sir. So, in the few slides that I am talking is already there in your parlance, and the public health is actively involved in this. I know beta thalassemia, Dharmapuri, Tiruvannamala and Kanyakumari district DDs will very well know that you are looking at this. But here again the effort is, we are looking at patients, but have you prevented them through genetic counseling, genetic preconceptional diagnosis? This is the role where we want the public health to work. Preconception diagnostics, preconception counseling, and preventing the occurrence of patients with all these diseases in these districts. Now you have sickle cell anemia in Nilgiris and Coimbatore districts. Alkaptonuria among Narikuravas in the Tirunelveli and Madurai zone. It's a huge problem. They don't come up to the public health or to the hospitals early. And with all the complications of alkaptonuria, they come in very late stages. That is how we first came to know that alkaptonuria is very common among these Narikurava tribes. But early diagnosis and preconceptionally, if you can advise them, we could prevent diseases and the cost. I will tell you one thing. Practically, everything boils down to the healthcare costs. The moment you have a complicated patient, you are spending 10 times more that you need to spend when you want to prevent a disease. Similarly, congenital heart disease, very interestingly, the national average is 2 to 2.43 per 1,000 live births. But in Tirunelveli district, the calculation is around 10 per 10,000 live births. We don't know what. The data is unclear. The public health here has a huge role. I will talk of some paradoxes. All persons with the BRCA1 or BRCA2 mutation, which you already have heard in terms of breast cancer, do not get breast cancers. Those who have mutation don't have breast cancers, and those who have breast cancers do not have the mutations. It is quid pro quo, it's both ways. Why is it? So why do certain people have risk? It is not because of these two genes. There are certain other complex combination of the genome, the transcriptome, the proteome, and the metabolome that is there in these patients, which needs to be discovered, and that could throw up a huge diagnostic and a risk factor assessment in cases where we want to find out breast cancer very early. Similarly, all obese patients do not have an acute coronary syndrome. At the same time, those who have acute coronary syndrome need not be obese or overweight. Here again, there is a underplay of a lot of other factors which we don't see in terms of their metabolomics, in terms of their proteomics, which will give us a huge understanding of the evolution of acute coronary syndrome, and that can help us to prevent acute coronary syndrome occurring by interfering in a young age, probably in the school level, and that could reduce the amount of money we are spending on our cardiology effort. Similarly, certain regions in our own state have people with similar racial characteristics developing goitrous thyroid with a high risk of thyroid cancer. But very interestingly, what we have seen is when the same racial group migrates to Bombay or Bangalore or Delhi, their risk of thyroid cancer decreases. When I say increased in risk of thyroid cancer in these communities, the first thing is probably something genetic. But when they migrate out, there is a decrease in risk, means there is something metabolic that is helping those people who are out of the community or out of the region. So, these regional racial issues are very important data for the public health to manage its interventions. Now coming to very important issue that we are going to see is the newborn screening. India did not have newborn screening till few years back. There was a pilot study that was done in Karnataka in Udupi where 6 disorders were taken, and the final report was very emphatic in saying that newborn screening as a public health effort is very much essential today because a huge cost is being spent in treating these children, in maintaining these children once the disease process has taken over the child. The statistic shows that 27,000 of our children yearly have intellectual disability because of errors of metabolism, which is preventable. And 2 lakhs and odd children have the threat of hemolysis and acute disease, or what you call as acute change in their metabolism, and even death. There are 5,000 odd children who go for adrenal crisis every time in this country because of the congenital adrenal hyperplasia. These are all issues now limited to the medical college, especially to the super specialty hospitals. But looking at this, you will understand this is an issue which the public health needs to be concerned. If you had found out these disorders early, the moment the child was born, if you had the capability of newborn screening when the child was born, you would have prevented many of these complications, and many of the hospitals who are treating this would have been empty today. But it is the endocrine department or many of the pediatric departments, especially the ICH in Chennai, which is busy combating this. Now coming to the final one, One Health approach. We are now looking at One Health approach as one major wing in preventive healthcare. Here you have the interplay of genomics, but more so an interplay of our own medical science, veterinary science, and of course the social and environmental sciences in one go, where you require the use of omic technologies, including microbiomics. This is a touch and go. I just like to mention for completion's sake what is personalized medicine. There is a time which has now come where every individual patient has a right to a particular scale of drug or a particular type of regimen based upon his own genomic or omic structure. I'll just give you a paradox. 2 patients with similar breast cancers with similar histology, grading and staging at the same age and weight have 2 dissimilar outcomes at the end of the same chemotherapeutic regimen. one person recovers, one person develops metastasis and dies. This is because of the genomics or the transcriptomics or the metabolomics are all together acting on the patient. If we had data, as a public health person, I can say this person has a good prognosis, this person has a good cancer or a bad cancer. The good cancer can be treated at a local level, the bad cancer requires some kind of a specific therapy. So this is one example where certain miRNAs are associated with good responses in lung cancers and bad responses, just a table to make you understand. Now coming to drug targets, I'll just pass it on fast. This scale of genomics or the omics technologies in the field will help you find out specific drug targets and drug development. The classical example is the vaccine we developed for COVID-19. Similarly, there are a lot of drug research that is happening for cancers which is based upon omic technologies. And of course, adverse drug reaction. I would like to show a very interesting slide, which is a newspaper report in 2006, which was called as the Chettiar problem. There is a very interesting thing is, before 2006, a lot of people from this community, including a few famous doctors, who went in for surgery and never recovered from anesthesia and died after 10 or 15 days of ventilation. That is because of a specific gene deficiency or defect which caused these people, now caused them not to recover from scoline injection. But today, there is a gene test, and we know that this population is at risk. This population awareness has been created, and now these people are being given a different drug for the same process. Now I have a couple of case studies in which I was also involved. In 2009, the neurologist in Tirunelveli brought in an interesting case of a 50-year-old person who had tremors, myoclonic jerks, and seizures. And we found out there was a clustering of cases in that part of the district. This is an effort which was taken in a medical college, but I will tell you, as public health experts, when you go into the community, there are small examples which are very similar to this that are there. Use of omics technologies would help you to track diseases, cohort the diseases, look at interventions. In 2010, we went in search of the first patient to a church in Tirunelveli, that is the Antonia Church in Puliyampeti. He was a 50-year-old man. When we asked for a clinical history, he said that almost 10 people in his family were already dead with this epileptic disorder. We went in search of patients and finally reported 726 individuals in 122 families who belong to a unique native South Indian community in those areas in South Tamil Nadu. And we had the help of the CSIR IGIB in terms of doing a genomic assessment of the rare disease. And finally, it was published in Brain, where we found that that entire community had a unique genetic mutation. And this is almost a 50 lakh population that is there in south of Tamil Nadu who can have this disease, who require specific drug treatment. And that drug treatment helps them in controlling the epilepsy and gives them a quality of life. So, this is where a tertiary care phenotype results in a population scale genomics and tracking down and cohorting of a disease. We came across another set of diseases where a 42-year-old agile man climbs up a tree in his home and was later found hanging dead on the tree top. A 17-year-old boy who was writing the exams just falls out of the table and dies. A 13-year-old teacher walking to the classroom falls down, drops down in front of the class and dies. Very interestingly, these are all sudden cardiac deaths, and all these three deaths happened in a particular area or a particular community in South Tamil Nadu. We went in search of these patients and we did 181 autopsies between 2010 and 2016, and very interestingly, 9.9% of the hearts in autopsy were normal in those dead people. And we continued the study between 2017 and 20. Of 92 autopsies we did, 11% of the hearts were normal. And a genetic analysis for these patients are ongoing. Very interestingly, this is a public health problem. Sudden cardiac death, this is a public health problem which requires an answer where omics technologies would help you. So the take-home message here is, Today, the benefits of omic technology is relegated to few individuals sitting in few departments in few medical colleges. But the potential of the technology is so much that it can improve the health and well-being of the populations. And so, at that point, we can even term it as public health omics. The promise of public health omics lays in analyzing relationships between the data and the health and disease state of populations, and that will help us in making critical decisions in public health. But the challenges that we have in omics is the cost. But once technology improves, the cost is bound to come down. So that is not an issue. But reproducibility of the data between different groups, the noise generated by the huge data set that comes out of the omics technologies, and the technical need to fish for novel markers, these three are major challenges for omics technologies used in public health. So with this short discourse, I stop here and finish with the point, what data that you are handling is like the tip of the iceberg. There is a lot more of data lying in public health in terms of technological diagnosis and technological findouts. The public health needs to rise to the occasion over the next 10 to 15 years. I think public health should have an armory of omics technologies to help you out to find diseases even before it can become clinically evident, and it requires a tertiary care intervention. We will be a happy and a healthy community if we can close our tertiary care hospitals someday in this country. Thank you so much.